Hello, and thank you all for coming to JoMarie Alba's virtual thesis defense presentation. This is the first time I've ever been involved in a thesis defense that is remote, where I'm sitting in my bedroom at home for the entire thing. So this is a really new experience for me, and I'm eager to get underway with it. So I am Kurt. I am JoMarie's major professor for these last 2 years as she is doing her thesis research, and I was the one that she was working with doing this work. And so I just want to say a couple words about her, and I'm really going to send it over to her for the rest of the time. One thing I do want to point out, though, just before we get going, is that there is, I think, up in the upper right hand corner, I believe, or left hand, I don't know which one I'm looking here. There is a little chat icon up there. During the course of the presentation, if you want to ask questions of JoMarie to answer at the end of the presentation, please go ahead, get in there, ask your questions, put your questions in the chat, and JoMarie can address those at the end of the chat. I'll be keeping my eyes on it too, so she doesn't miss anything or, you know, inexplicably there. So JoMarie came to us from Southeast Alaska. She had grown up much of her life there and did her undergrad at University of Alaska Southeast. And then I met her the way I meet most people these days, it seems like, by email. And it was 2 years ago, about this time, that I was emailing back and forth with her about her impending trip down to Walla Walla, Washington to begin her master's program with me. And it was about that time also that she let me know that she had just received a graduate research fellowship from the National Science Foundation. And at that time, I was actually kind of wondering whether or not she was going to say, well, see ya, that was really nice, but now I can get into a lot better schools than Walla Walla. Honestly, we don't have a lot of people who get GRFPs end up coming to Walla Walla University. So I was very excited that she still wanted to come and work at Walla Walla University. So I'm glad she was here. And especially because while she was here, she actually had to deal with a lot of things. Like, there was a lot of adversity that was kind of thrown at her during the course of her master's here. For instance, one of them, our school, Walla Walla University, and the NSF just do not seem to like mesh that well. And so she's had these problems of her GRFP fellowship just not getting paid out to her for months and months and months on end, and has had to deal with that. Also, she has had to deal with a semi-absent professor because I became director of a reservation marine lab in the middle of this. I had no idea what I was doing with that. During the summer that she was doing her research, I actually was not able to dive with her once to help her collect her octopuses. And so she had to deal with that as well. But despite all those challenges, despite all that adversity, JoMarie tackled all of them with a real amazing amount of character. And I've been really proud to see her grow through that, grow as a scientist while she has been here. And I'm just really excited to see her at this point where she is now presenting her thesis research, coming to the end of this stage where she has a really good product. I'm really excited for her to tell you all about. So with that, what I'm going to do is I am going to now send it over to JoMarie to tell you directly about these things. And yeah, if I can get this to work, hold on here. There we go. And here you are, JoMarie. You're muted.
All right, can you hear me now? Perfect. Thank you everyone for being here. I'd like to say that I love octopus behavior. I'm fascinated at their ability to change colors instantaneously. I love that they're immensely clever and they can learn through observation. I love that they can teach themselves how to open up jars, cork jars, to obtain their favorite food item. And I also love the fact that they're really good at escaping aquaria. And so it's really concerning for me that humans have essentially changed the environment where octopuses live through ocean acidification and warming. And I began to question how exactly these climate stressors are affecting octopus behavior, specifically their predatory behaviors. How is that changing? Because it made me think, you know, are they changing the way they eat their food? Is that going to lead to a trophic cascade? Is the marine ecosystem going to crash? Are we going to have a change in biodiversity? And it's that question, how do climate stressors affect the predatory responses of octopus rubescens, that has been the catalyst of my thesis research. To start, I want to talk about ocean warming and ocean acidification. Since the Industrial Revolution, the concentration of carbon dioxide has increased from 280 parts per million to over 400 parts per million. Last year, the average measured carbon dioxide concentration was 416.21 parts per million, and that is a 50% increase in relation to the start of the Industrial Revolution. And this is concerning because CO2 is a greenhouse gas, and greenhouse gases cause the greenhouse effect, where greenhouse gases act as an insulating layer and trap infrared heat, thereby increasing the temperature of the planet, also increasing the temperature of the ocean, ultimately leading to global warming. How much has it risen since the end of the 19th century? Well, it's risen 1 degree Celsius. And if we keep business as usual and do nothing to curb our fossil fuel usage, the most extreme end of the century climate prediction, known as RCP 8.5, predicts that the temperature is gonna increase 3.7 to 4.8 degrees relative to the 19th century. The ocean, carbon dioxide is also changing the ocean in another way by decreasing the pH. And in order to understand this, you need to know that the partial pressure, that the air and the ocean are in gas equilibrium, which basically means that the partial pressure of carbon dioxide pushing down onto the ocean is equal to the partial pressure of the carbon dioxide in the ocean pushing up into the air. Since we've increased the amount of CO2 into the atmosphere, it's creating a larger amount of pressure being pushed down into the ocean, thereby increasing the amount of CO2 in the ocean. So what's exactly happening once CO2 is in the ocean? It reacts with water and forms carbonic acid. And carbonic acid will dissociate into a bicarbonate ion and a hydrogen ion. This bicarbonate ion will further dissociate and break down to a carbonate ion and a hydrogen ion. And it's this hydrogen ion that we should be concerned about, because as the amount of hydrogen increases in the ocean, it leads to a decrease in pH, ultimately leading to ocean acidification. How much has the pH changed since the Industrial Revolution? It's dropped from 8.2 down to 8.1. And based on RCP 8.5, it predicts that the pH will drop down to 7.8. And this may seem like a small value, but I'd like to remind you that that pH is an inverse exponential relationship with the concentration of hydrogen. And when you calculate the numbers, and I provided the equation here, you'll see that there is an order of magnitude difference between 8.2 and 7.8. But what exactly does it mean to have an order of magnitude difference? Well, if you think about it in terms that we all understand, money, I think you'll get it. Because if you found out that you had won $100,000, you'd be ecstatic. But then if you found out that you had won $1 million, you went from ecstatic to just, oh. But if we switch that around and you found out that you were in debt for $100,000, I think you would change your life choices and kind of figure out what you need to do to pay off that debt. But then if you found out that you had $1 million in debt, well, I don't even know what you'd feel like. But you can, with that example, you can understand what an order of magnitude a change in order of magnitude can mean, especially in terms of hydrogen concentration in the ocean. So changes in ocean chemistry and changes in ocean temperature can affect marine animals like cephalopods. And research has started to show that changes in physiological performance can lead to changes in behaviors. And we're now beginning to understand that these changes in behaviors lead to differences in species interactions, like predator-prey interactions. And species interactions are important because they contribute to the shaping and function of marine ecosystems. And there's a growing concern that when a marine ecosystem changes, it's going to be detrimental to some species and beneficial to others, which can ultimately lead to changes in biodiversity. So small changes in behavior could have far-reaching implications that we have yet to understand. Okay. So why study cephalopods? Cephalopods are ecologically important. They're preyed upon by many large marine animals. They are predators, they are predators to many marine invertebrates and fishes, and thereby they link trophic levels and food webs together. They're also economically important due to their growing presence in global fisheries. And finally, because of their rapid growth, short lifespan, and behavioral plasticity, they're highly adaptive to changing environments. And this adaptability may give them an advantage over other species in the face of climate change. Oops, excuse me. So most of the studies looking at climate change and cephalopods have primarily focused on physiological traits. Of these studies, only 3 have investigated cephalopod behavior in response to ocean acidification, and only 2 of these studies have looked into predator-prey interactions. We still don't know how these physiological responses and behavioral changes are affecting marine ecosystems and biodiversity. But what we do know is that that cephalopod numbers are growing worldwide, along with that their range is expanding. Since climate change is shifting environmental factors, it can alter the predatory strategies of cephalopods. And understanding the predatory responses of cephalopods may be helpful in forecasting the impacts of ocean change, but we first need to obtain baseline information of the responses to climate stressors. Therefore, the objective of the study is to understand how ocean warming, acidification, and their co-occurrence affect the interactions between octopus rubescence and its prey. And to meet this objective, I asked 2 research questions. The first is, does ocean warming, acidification, and their interaction affect the predatory strategies, used by octopus rubescents to consume Hemigraphis nudus, the purple shore crab? Does ocean warming, acidification, or their interaction affect the octopus rubescent's ability to drill holes? And I hypothesize that ocean acidification, warming, and their interaction will cause a difference in predatory strategies and drilling ability. And I predict that these differences will negatively impact octopus predatory behaviors. Now, how do I answer these questions? Well, let me show you. First, I had to collect octopuses, and Walla Walla University has an awesome marine station located on Fidalgo Island, and south of our marine station is our octopus collection site. And in order for us to collect octopuses, we have to go scuba diving. And from the shore, we dive down to the bottom of the ocean. From the shore, we dive down from the bottom of the ocean, and located on the bottom of the ocean are discarded glass bottles. And why these bottles are here, we're not sure. But octopuses have taken advantage and utilized these bottles as dens. So we swim around, look through these bottles, and see if we can find octopuses. And when we do, we collect the bottle, place it in a bag, and take it up to the surface. And once at the surface, we then transfer the octopuses out of the bottles, as you can see here, into red plastic bottles. Sometimes this happens quite easily. Other times it does not. Eventually, we get all the octopuses into these red plastic bottles, and we stick them in a cooler filled with seawater, and we place them in the back of my van, and we drive back to Rosario Beach Marine Lab. Once there, I collect octopus intake data, which involves obtaining the weight and the sex of the octopus. Once I do that, I transfer the octopus into enclosures to allow them to acclimate to laboratory conditions. And these enclosures, as you can see here, are connected to the ambient flow through seawater system at Rosario Beach Marine Lab. And these octopuses will acclimate for about a minimum of 1 week before I transfer them into the ocean acidification system. So the ocean acidification system that we utilize in this experiment was custom built by previous grad student Monica Culler and Dr. Kurt Onthank. And here is a modeled rendering of the ocean acidification system. You'll see here the holding tank and a pump that is connected to the tank, which draws out water and pushes the water through a chiller, which pushes it through a Venturi injector, which reoxygenates the water with CO2-free air. So the water that gets pushed back into the tank is at the perfect temperature and is freshly oxygenated. What I forgot to mention is that this tank is a temperature-controlled, partially closed recirculating system, if you haven't figured that out yet. Attached in the top of the tank are 2 probes, a temperature probe and a pH probe. And these 2 probes provide input to the control stat box to control either the temperature of the tank or the pH of the tank. If the temperature of the tank gets too warm, the chiller will turn on. And then once it reaches the ideal temperature, the pH control stat box will turn off the chiller. The pH probe measures pH of the seawater inside of the tank, and if the pH gets too high, it tells the control box, which turns on a solenoid valve, which will bubble in CO2 gas until the ideal pH is reached. Modifications to the system include the addition of inline external heater, and which I added in between the chiller and the Venturi injector. And by adding this, we also had to update the software to include a heat function. And here is a picture of our ocean acidification. You'll see the control stat boxes and the different OA acidification systems. All of the control stat boxes are connected to the internet. So we're continuously collecting temperature and pH data, and we have the ability to check in real time what the pH and temperature of each tank is. Now, going back to the octopuses who are sitting in the acclimation tanks waiting to be placed in treatments. The different treatments that I created, that I determined were either control or projected microatmospheres of PCO2 combined with either control or projected temperature, the control or projected temperature. And in ocean acidification research, we measure carbon dioxide through microatmospheres of the partial pressure of carbon dioxide, which is a mouthful to say and kind of hard to understand. So I've provided the corresponding pH values here. Okay, to see what that looks like, we have the first treatment, which is control PCO2 with controlled temperature. The next treatment is projected PCO2 with controlled temperature, followed by control PCO2 and projected temperature, and finally projected PCO2 and a projected temperature. Now, you may be asking, how did I determine the control CO2 treatment? Well, while scuba diving, I also collected seawater samples and returned them to Rosario Beach Marine Lab, where we determined the temperature, the pH, the alkalinity, and the salinity of every seawater sample. And then we use these measured values and plug them into a function in the CCAR package to determine the PCO2 of the seawater sample. And we found that the average PCO2 at the octopus collection site was around 800 microatmospheres. The projected treatment was based on the RCP 8.5 predictions, leading me to determine a projected PCO2 value of 1,350 microatmospheres. How did I determine the controlled temperature? This was a little bit easier. We contacted Northwest Environmental Moorings, who maintain a bunch of CTD moorings within the Salish Seas. And we selected a mooring that was closest to our octopus collection site and downloaded the data. And the data that we got ranged in time between November 2005 to May 2019. And I found the average temperature between the months of May and September, which were the months that I'd be conducting the experiment, and finding the temperature at the depth of 10 to 20 meters, which were the depths that we'd be locating and collecting octopus rubescents. So the average temperature was 12.1 degrees Celsius. And again, utilizing RCP 8.5, I obtained a projected temperature of 15.8 degrees Celsius. Okay, so we have octopus sitting in acclimation tanks. Our treatment is ready. Once I have the ocean acidification systems up and running, I randomly place the octopuses into one of the 4 treatment tanks. And then during treatment, I fed them manila clams. And the octopuses stayed in treatment for about 2 weeks. And after the 2 weeks, I fed the octopus a purple shore crab. And this kind of leads us into our first experiment, our first question of, also known as the predatory behavioral trials. And the predatory behavioral trials tries to answer this question: does ocean acidification, warming, and their interaction affect the predatory strategies used by octopus rubescents in capturing Hemigraphis nudus? So the predatory behavioral trials are just components of the octopus's visual attack sequence. Excuse me. And the visual attack sequence of octopuses are detect, approach, pause, attack. So the octopuses have now been sitting in treatment for 2 weeks. I fed them a purple shore crab, and I recorded the interaction between the octopus and the purple shore crab with a GoPro Hero 7 that was attached to a custom-built mount that sat on top of one of the ocean acidification tanks with the lid removed. Once I recorded all of the videos, I utilized the program's VLC Media Player and Fiji's ImageJ to extract the data. So the behavior parameters, the predatory behavior strategies that I measured are striking distance, which is the distance between the octopus and the crab before it attacks. Latency to attack, which is the amount of time the octopus waits before attacking the crab. Predator to prey orientation, which is the orientation of the crab to the octopus. And if we have this imaginary crosshair right here, and 0 degrees is straight in front of the octopus, where is the crab in relation to 0 degrees? Next, there's type of attack. What kind of attack did the octopus use to capture the crab? Did it use a ballistic pounce where it jets through the water, opening up its interbranchial web before capturing the crab? Or did it use an arm grab where all it did was reach out with its arms to grab the crab? And finally, the last predatory behavior parameter was body pattern. What was the body pattern that the octopus exhibited while attacking the crab? Once I extracted all my data, I utilized the statistical software R to conduct a 2-factor multivariate analysis of variance and a Cochrane mental Heinzel test. Now, I'm going to talk you through this figure because the next 2 following figures are similar to it. Oop, okay. Here on the x-axis, you have the CO2 treatments, the left side representing the control and the right side representing the projected CO2 treatment. The color of the outline of the boxes represents the temperature treatments, where this blue color represents the control temperature and the orange color represents the projected temperature treatment. Here on the y-axis is the predatory behavior parameter that I measured, and in this one, it's latency to attack measured in seconds. Now, with this box and whisker, the whiskers represent the minimum and the maximum, and the box represents 50% of the data, and this line represents the median. Now, what I'd like to point out is that there is a wide range of variability within each treatment group, and that this variability overlaps with one another. So when you see something like this, you can guess that the statistical test is not significant. And we confirm this with the two-way MANOVA, showing us that the p-values are not 0.05. Any p-value that's less than 0.05 indicates a significant test. but none of these are less than 0.05. Next, we have striking distance. Again, we have CO2 treatment on the x-axis. The colors of the boxes represent the temperature treatment. And then this time on the y-axis, we have striking distance, which is measured in centimeters. And I'd like to point out again, the wide range of variability that exists within each treatment and how they overlap amongst treatments. And conducting our test, we can see that the p-values are all greater than 0.05. So this test is also not significant. Next, we have orientation, predator-prey orientation, measured in degrees. X-axis stays the same. Colors of the boxes represent the same thing. Again, you'll see wide range of variability that exists and overlap. And looking at our p-values, we can see and confirm that the effects of ocean warming and acidification do not significantly affect this predatory behavior. Up next, we have a bar plot representing the frequency of octopuses who performed a certain behavior. So this is type of attack. What type of attack did the octopus use to capture the crab? Here on the x-axis, you'll see the CO2 treatments. Notice that it now is control, projected, control, projected. On the y-axis, we have frequency. And up top, we have the temperature treatments. These two bars over here represent the control temperature, and these two bars over here represent the projected temperature. Our legend here tells us that gray represents the octopuses who used a pounce, and that the orange represents the octopuses who used an arm grab. And as you can see, that's a lot of gray. And when you look, and we can confirm that there's no difference amongst treatment groups because our p-value is 0.95, which is almost 1. So ocean acidification and warming does not affect the type of attack an octopus uses. Next, we have body pattern. X-axis is the same, y-axis is the same, the separation between temperature treatments is the same. But now we have these colors corresponding to the body pattern that the octopus used while attacking the crab. We have blush, gray, and red. And superficially, when you look at this graph, you don't really see any type of pattern. And when we conduct the Cochrane mental Heinzel test, we obtain a p-value of 0.69. So this test is also not significant. So the results of this first experiment indicate that elevated CO2 and temperature had no measurable effect on the predatory behavior strategies, latency to attack, striking distance, predator-prey orientation, type of attack, and body pattern of octopus rubescence. And having no significant results is just as exciting as having significant results. It's all a matter of how you interpret it. Now that I've answered that first question, I want to go back and show you how I answered that second question about drill hole ability of octopuses in the face of climate change. But before I do that, I want to talk about point patterns. You'll hear me throughout the presentation utilize the words point patterns, spatial patterns. I use these terms interchangeably. And a point pattern is a pattern of points. And there are three kinds. There are clustered, there is random, and there is regular. And each of the three boxes that you see here represent a different type of point pattern. And I wanted to ask you to look at these boxes and see if you can tell which box is which one is clustered, which one is random, which one is regular. And I'll give you a few seconds to think about it. So this first box is clustered, and you can see that's it's quite obvious. The dots in a clustered point pattern influence the dots surrounding it by being closer to one another. The dots are dependent of one another by being closer to one another. And these next two, this one is random, the middle one is random, and the one on the right is regular. You can see that it's kind of hard to tell the difference between the two. But if you look closely in this middle box, you'll see that some dots, some points overlap one another. And within a random distribution, the points do not depend on one another on where they're located. And that's how you can end up with point patterns that are clustered on top, or I'm sorry, that are overlapping one another. But if you look at this regular pattern, you'll see that none of the points are overlapping. And that's because in a regular pattern, the point influences other points by ensuring that there are certain distances away from one another. Okay, switching gears, I'm now going to talk about octopus drilling behaviors. So octopuses are unique among cephalopods because they prey on organisms with strong defense mechanisms like this clam and this crab. Now let's think about a clam. When an octopus wants to eat a clam, he faces the problem of having to try to get into the shell to collect the visceral mass. So what the octopus will do is it'll use its arm and pull the clam apart. And if the pulling action fails, the octopus will switch strategies and drill a hole into the shell of the clam. How does it do that? Well, the octopus, as some of you may know, they have a beak, and inside of that beak, they have two appendages. The radula, which is its scraping tongue, and the salivary papilla. And the octopus will use the salivary papilla to drill a hole into the shell of its prey, and will continue to do so until it penetrates through. And once it does, it will inject a venom which paralyzes the prey. Therefore, the muscles that are holding the clam shut become paralyzed and open up, enabling the octopus to reach into the shell and consume the visceral mass. Another fascinating thing about octopus drilling behavior is that they can do selective drilling, which you can see in this figure here. You have two valves of the same shell, and the drill holes are represented by these tiny dots. What you'll notice about these tiny dots is that they congregate around these large circles, and these large circles represent the adductor muscle attachment. So what this image is showing you is that octopuses have the ability to locate the muscle attachment to drill a hole and inject the venom so that it can paralyze its prey. How does the octopus know where to drill? We have no idea. But something else that I'd like to point out is if you look at the anterior adductor muscle And since this is a clam, here it's its umbo, and the umbo points forward. This is its adductor muscle attachment. And if you look superficially, it looks like there are more dots, more drill holes located on the adductor muscle. Why does the octopus do this? We don't know. What we do know is that octopuses can do selective drilling locating the adductor muscle. So knowing this, I wondered, and I wanted to ask the question, how does warming, acidification, or their interaction affect an octopus's ability to drill holes? Basically, do climate stressors make octopuses worse at locating the adductor muscle? So in order to collect octopus drill hole data, if you can remember, I fed the octopuses clams while they were in treatment, and every day I collected discarded shells and photographed shells with drill holes, and I standardized these images utilizing a new image manipulation program and G3 data, which is a data extraction program. Okay. Before moving on, it is imperative for you to understand Ripley's L function. However, I'm not going to explain the equation or the function. I'm just going to explain to you how to interpret the results of a Ripley's L function. What you need to know is that the expected Ripley's L function is the expected spatial pattern of a Ripley's L function is always going to be random, which is represented by this black line down the middle. And the observed L function is represented by this red line. And this number is calculated with using Ripley's L function. And the important thing to remember is that any observed value that is below 0 represents a regular spatial pattern. Any observed value that is above 0 represents a clustered spatial pattern. And this regularity or clustering is significant if the observed line is outside of the bounds of the confidence interval envelope, which is represented by this gray area here. So at smaller values of R, we have a regular point pattern, and then at larger values of R, we have a clustered point pattern. So what does this look like? It looks like this. If you look at an individual dot, at an individual point, you'll see that as you grow a circle larger and larger, that the points closest to that individual dot exhibit a regular point pattern. But then if you increase that circle around the individual dot, you're coming across a lot of empty spaces and then a bunch of clustered drill holes. So this point pattern is represented by this Ripley's L function. So for this part of the experiment, I wanted to know if climate stressors affected the ability of how an octopus drilled a hole. And basically, do climate stressors make octopus worse at locating the adductor muscle, which we can measure by measuring how tight or how loose the variability between the clusters is. So we developed this function that we deemed M of R, where it compares the L function of one group against the L function of the other group, and it determines relative clustering between these 2 point patterns. In order to do this, we needed to first determine, well, are the point patterns on the clamshell clustered? Yes, as you can see here, after what you just learned about Ripley's L function, that the observed data above is significantly above the confidence interval envelope. So again, here is Ripley's L function. It's just here for you to visualize. We take the L function of one group, say control CO2, and we minus it with the L function of experimental or projected CO2 and compare what the variability is and see between the two which one is more clustered, tightly clustered, and which one is more loosely clustered. Okay, so there is a significant difference between, I shouldn't use that term, there is a difference between the L function figure that I showed you and an M function figure. And before, where any observed value of L above the 0 represented a clustering pattern and below 0 represented a regular pattern, with the M function, it changes to where any value above 0 represents a tighter clustering in the first group, and any value below 0 represents a tighter clustering in the second group. So here we have the drill hole patterns based on CO2 treatment. Purple represents the control CO2, orange represents the projected CO2 treatment. And you'll look at these drill holes and they're just kind of intermixed with one another. And looking at the M function graph, you can see that the observed data at multiple distances of R never escapes the bounds of the confidence interval envelope. So this means that CO2 does not affect the drill holing abilities of octopus rubescens, and we obtained a p-value of 0.08 to confirm this. What about temperature? All right, so this time purple is represented, purple represents the control temperature and orange represents the experimental projected temperature. Again, you'll see on the clam that the drill hole patterns are kind of intermixed with one another and hard to really separate them apart. And you look at the corresponding M function graph and you can see that maybe at very small values of R that the higher temperature leads to a tighter clustering of drill holes. However, throughout distances of R, it's not significantly different. And then when we conduct our test, we obtained a p-value of 0.47, telling us that temperature does not affect the drilling abilities of octopus rubescens. So thus far, ocean acidification and the temperature has not affected any predatory behavior parameters or drilling abilities of octopus rubescens. So we wanted to ask, well, what about the side of the clam, regardless of what the temperature or CO2 treatment is? Now you'll see here that purple represents the drill holes located on the anterior end of the clam, and orange represents the drill holes located on the posterior end of the clam. And if you look at its corresponding M function graph, you will see that the drill holes located on the anterior end of the clam seem to be more tightly clustered compared to the posterior end of the clam. And we can confirm that they're significantly different because we obtained a p-value that's less than 0.01. Woohoo! first and only significant result. And this is exciting for us because as I told you, we don't know much about octopus drill hole behavior other than the fact that they do it with their salivary papilla. And we know that they can utilize, that they can locate the muscle attachment in their shelled prey. But we don't know how they know, and we don't know why they prefer the anterior end. But the piece of the puzzle that we've added is that we now can show that on the anterior end of the clam, the drill holes are more tightly clustered compared to the posterior end. And this is a small discovery, but a discovery nonetheless, and we're hoping to write a manuscript and send it off to publication. Fingers crossed. Okay, so in conclusion, the non-significant results indicate that elevated warming and acidification will have minimum effects, if any, on the predatory behavior and drilling ability of octopus rubescents. So what does this mean? Well, it is possible that due to an octopus's adaptability and plasticity, that octopus rubescents may be resilient to future ocean conditions. It is also possible that the power to see the effects were reduced by that intense individual variability and the small sample size. But regardless of the absence of evidence in the predatory behavior parameters and regardless of the evidence of how ocean acidification and warming affect the predatory behaviors and drilling abilities of octopus rubescents, this study inspired the development of a novel function, MR. And I hope to shift the current paradigm to utilize more robust statistical methods to analyze octopus drill hole patterns. So with that, before I take questions, I wanted to say many thanks. First to my funders, National Science Foundation. Many thanks to the biology department at Walla Walla University and to those at Rosario Beach Marine Lab. I'd also like to thank Dr. Kurt Onthank. I really have no words to express the gratitude that I feel towards Dr. Onthank. He is an awesome major professor to have, especially for conducting a thesis experiment. You and Stephanie and Ruby and Lakeland and Rowan have not only welcomed me into your lab, but you also welcomed me into your family, which made the transition to Walla Walla a lot more smooth for me. I'd like to take a moment to thank Dr. CJ Brothers and Dr. Jim Nestler, who are on my committee. Those two have gone above and beyond what I expected out of committee members by driving many long hours to go scuba diving with me. And Dr. Kyle Craig, who is an observer of this process. Thank you very much. And of course, all the biological sciences faculty at Walla Walla University. I want to say many thanks to Onthank lab members, past and present. You guys have been awesome, and I'm really glad that we've gotten to know each other in this short amount of time. And I really would like to shout out to this guy right here, Jared Sexton. He was my undergraduate assistant over the summer, and he is like the best assistant ever, and I'm immensely grateful for the amount of help that you provided this past summer. Many thanks to my friends, hello, my previous mentors, hello, my parents, thanks mom and dad, my sisters, Tiffany and Crystal and their babies, my giant family, and of course, this guy right here, my husband, Max. He has been immensely supportive for all the crazy ideas that I have, and he's been the best adventure buddy, and like the past 2 weeks has been great by ensuring that I eat and making sure that I go outside and making sure that I get all my work done. And so finally, I'm done, and I will take any questions. Oh, and if you have Twitter, you can tweet me your questions at JoMarie Defends. Thank you.
So good job, JoMarie. So here's what happened is I messed up making the Q&A. And so that was a problem. Hold on here. So this is what I'm going to do. I have a question here that I will ask you from Callista here in a minute. But if you have questions, send your questions to my email, because I totally forgot to set this up. So if you have questions, send them to my email, because I totally forgot to, or messed up setting up the Q&A. So you can't find it. That's totally my bad. Send them to my email. I'll ask them of JoMarie. But in the meantime, I have one that had come in already. So JoMarie, Callista asks, did the octopus ever ignore the prey? And if so, hold on here. And if so, how did that factor into your calculations? I'm sending you live again here. You're muted.
Did the octopus affect or ignore the prey? Yes, they did. So what I did with my data is basically I had to separate it out, and initially I collected, I think, 31 or 32 octopuses, and of those octopuses, I used 29 of them in the experiments, and of the 29, only 21 of them ate a crab. And so what I did was basically I repeated the predatory behavioral trial. So if an octopus did not eat a prey, I went back the next day, and I had to do this with all the octopuses, and fed him another crab and recorded that interaction. And I did it a third time to make sure that the behavior that they were exhibiting was consistent. And basically, if an octopus was consistent and not eating a prey, we just said, okay, he didn't eat one. We're just going to not count you within the data analysis with the predatory behavior trial. So hopefully that answers your question. Was there more to it?
Hold on. No. I think that was it. So I have another one from Jim Nessler sent in another one. So here's Jim Nessler's question. Median orientations all seem to be close to zero, suggesting that most animals are directly facing the prey before they attack. Did the octopuses rotate themselves to face prey prior to attack? And then secondly, did the octopuses wait until the prey moved in front of them before they attacked? Okay, so here, I'm going to get you all queued up here again. Send you back live.
So the question was, did the octopus move to orient itself to the crab? Is that correct? Yes. Okay. Yes, it did. Based on so the octopus typically approached the crab 3 different ways. It would either jet and then land and then pause. If it did that, it typically just stayed in that position before it attacked the crab, stayed in that orientation. And sometimes when the octopus approached the crab through crawling, while crawling and making its way approaching to the crab, it would change its orientation and then pause. And the orientation that I used was really at that pause before the octopus initiated the final attack. Okay, there was a second part to the question and I forgot what it was.
The second part was, or did they wait for the prey to move in front of them first?
Oh, yes, they totally did. Sometimes octopuses would literally wait for the crab to just crawl right in front of them, and they would just grab it or jump on top of it. Other times, octopuses would jump the length of the, they wouldn't wait, and they would just jump. And I don't know why they exhibited like these changes in behavior, like why there was that variability in how much they waited. It's just kind of, you know, it could be a personality thing, because octopuses do have personality. It could just be like, where were they and like how hungry were they? Did the treatments, you know, affect them in any way that made them act this way? But based on my statistical tests, I wasn't able to detect any of these differences. But to go back and answer your question, yes, octopuses, depending on the octopus, they would wait until for the crab to move right in front of them. It did happen.
Okay. Okay, I have another question here from Sherry Tamoni. She asks, how comfortable are your octopus at the time of your experiments? Were they acclimated to your tanks and crabs? Is crabs their favorite thing to eat?
So the question was how comfortable? Is that right?
Yes. Yes.
I'm not quite sure what you mean by how comfortable, but we do know with metabolic studies conducted in this lab that it takes about a week for an octopus to acclimate to new situations in a physiological and metabolic standpoint. So I kind of based on acclimation period, like on that study. It's just like, okay, cool. If they're comfortable in treatment after a week, maybe they'll be comfortable in acclimation after a week. And to answer your question, yes, octopuses love crab. It is their favorite thing to eat, which is why I chose to feed octopuses the crab, because in my head, I naively thought, oh yeah, if it's their favorite food, they're obviously going to attack it every single time, right? But of course, when it comes to behavior studies, whenever you want an animal to perform a certain behavior, it won't do it, which totally happened with my research.
Okay, got another one here from Ricky Wright. He asks, while your research didn't uncover many solid correlations, do you think that there are some definite effects on octopuses that you couldn't detect? Hold on, I'm gonna get you back here.
Oh yeah, okay. So that could also be like the reason why I didn't detect anything was just like how I was measuring these changes. And to answer your question, yes. You know, when I created these parameters, they were based on a previous research that found significant differences in the predatory strategies of squid species. And I thought, oh, okay, I can just like transpose these things that he's measuring to an octopus. They're closely related, right? But they're completely different animals that exhibit completely different behaviors. So like it could totally be possible that the reason why I didn't see the effects of ocean acidification and warming is because I was measuring things the wrong way. I was measuring the wrong thing the wrong way, or I could have been doing a statistical analysis the wrong way. You know, I could maybe like, if my sample sizes were bigger, maybe take all my continuous variables and like lump them into categories. But c'est la vie.
Okay, got another question here from a few questions from Warren Carlisle. I'll ask you one of them because we have a lot of questions coming in. He asked, with the octopuses that passed, were there rulings of death? Hold on here, get you back up.
I'm confused by the question.
I think he's asking why did they die?
Why did who die? The octopuses?
The octopuses.
Did I mention that octopuses died?
Let's go on to the next question.
No. I mean, like,
okay, he also
one of those things like,
any
how did you know they died? I didn't say. But to just tell you a quick story about octopuses dying, yes, they do die while acclimating to the lab, and either acclimating to lab conditions. I had one octopus die while in treatment. I don't know why.
He also asked, did any stop eating altogether?
Did any of them stop eating altogether? Yes. Some octopuses, while in acclimation, were eating just fine. And then when I put them in treatment, they didn't eat at all. And the only thing that I could think of was maybe placing them in treatment exacerbated the senescence process. It's possible that the octopuses were, the ones that I brought back, were really old and were going through the senescence phase, which is basically an octopus's slow phase of dying. And while in acclimation, they were eating just fine. They look super duper healthy. And then I'll put them in treatment, which stressed them out, which may have sped up their process of going into senescence. And during senescence, they don't eat. So that's a possible explanation, but I don't know if that's true for all of them. Maybe some octopuses are just really good at starving themselves for 2 weeks too. Because there were situations where none of the octopuses would eat for 2 weeks, and I took them out of treatment and put them back into the acclimation tanks before releasing them back to the octopus collection site. And while they were waiting to be released, they started eating again. I don't know why.
I'm going to see if I can just I can ask you questions without actually putting my picture up there, because I don't think anyone really wants to look at me. So next up question. You can understand me, right, JoMarie?
Yeah,
can you hear me?
You can say the question, and I can repeat the question.
Just in case other people can't hear. Yeah, okay. So Jenny Hoffmeister asks, in your experiment, the clams didn't experience a low pH environment. So do you think results would change if the clam was also under the stressors of high temp or low pH?
Yes, I think the results would change as well. A reason why we didn't do that was because we were limited in ocean acidification systems, which is really the case for all ocean acidification studies. However, after conducting the study, I kind of realized I should have, in a perfect world, hindsight's 2020, I should have placed the prey items in acidified or warm treatment. But in order to do something like that, I think I would need a larger sample sizes of octopuses, because not only do you now have 4 treatments for the octopuses, you're also adding the 4 treatments of the prey species, which leads to a combination of 16 different combinations, something like that, which would be crazy, and that sounds super complicated. But at the end of the day, I do think that introducing prey species to acidification or warming would lead to a different response.
Okay, another one here. Hold on. Okay, so this one is from Lloyd Trueblood. Does Octopus rubescens only drill one hole when it handles a bivalve, or were there multiple drill hole attempts? And so if there are multiple attempts,
okay, I was going to repeat.
Okay. Oh, actually, I think people can hear. Steph said that she can hear me as well, so I think people can hear me.
Okay. Perfect.
Yeah. So does Octopus rubescens drill only one hole when it handles a bivalve, or were there multiple drill hole attempts? And if there are multiple attempts, is there any effect of temperature or OA in the number of drill hole attempts per prey item?
Ooh, that's a good question. Okay, to answer your question, yes, there were some octopuses who drilled multiple drill holes on the clam. Some of them were attempted drill holes and didn't drill all the way, and some of them were like multiple completed drill holes. What was the second half?
If there was multiple attempts, did you see differences in the number of attempts per treatment?
I didn't check. I have that information. I haven't checked, but I will, and I'll let you know. Let me get back to you, Lloyd.
Carolyn Bergstrom asks, okay. Would you explain what the distance R axis represented again? While non-significant, it looked like there was a consistent trend for the heated treatment to lead to more clustering of the attacks on clams.
Hold on here. So Carolyn is asking what the distance means.
Yes. What was the distance, the R axis, your X axis on your figures? What did that represent again?
Oh, okay. Of course, Carolyn, you would ask a question like this. Ken, I'm going to share my screen. Hold on.
Let's do that.
Take a second. Up. Okay, hold on a second, Carolyn, as I try to explain this in a way that you can understand through pictures. Whoops. Whoops. Apparently I'm failing at it. Oh my goodness. So basically R represents the distance of, it represents radius, which is the area of the circle that surrounds the individual point pattern. And I'm trying to load a PowerPoint while speaking to you. And hopefully that will do this, but it looks like my computer might freeze. So maybe you can draw it. Can I do this? Here, desktop. Oh no, this isn't working. I'm so sorry. Oh well. Okay, it's not working. So maybe I'll draw it. Okay, you have, whoops, different dots within a spatial point pattern. And Ripley's L basically looks into how many point pattern pairs occur within an area of the circle. So say you have, whoops, this dot right here and you try to count how many other dots are within that circle. Right now there's 0. So you increase the radius of the circle and now count how many other events are occurring in this area of the circle. And you slowly increase the radius of the circle until all dots are pretty much in one circle. So the idea behind this is that as you increase the distance of R, you get an exponential growth of the amount of points that occur within the area of the circle. And when the data is random, when there is a random spatial point pattern, you would expect that the point density, which is the number of points over the area. No, I'm confused. Anywho, I feel like this would be a great conversation that you and I can have where I'm not awkwardly trying to draw this on a piece of paper. But basically, as you increase the area of R, you're increasing the area in which the test is counting how many other points there are and comparing it to the random distribution. And it's clustered when the point density is greater than random, and it's regular when the point density is less than random. I hope that kind of sort of answers your question, and maybe this was funny and entertaining for people to watch for me to try to answer that. Okay, next question.
Okay, from Jared Sexton, he asks, at the end you mentioned hoping to shift the paradigm about how we study octopus drill holes. Could you say more about how you're hoping this changes and how we could do more to study what's going on here in greater detail?
Okay, sure. So this study is the first to use multi-distance spatial cluster analysis to discern the point patterns of octopus drill holes on their prey. In the past, all previous research looking into octopus drill hole patterns utilize some form of, they utilize the chi-square test. And the chi-square test, as some of you may know, is, it's really easy to do. You can do it by hand, but it's limited because you have to divide the region of interest into different sections, and the test is highly dependent on the size or the number of these sections, and these sections are artificially partitioned by the researchers. So in addition, chi-square tests don't have the ability to detect regular point patterns. It can tell you if something is random, and it can tell you if something is clustered, but it can't tell you if something has a random point pattern. So utilizing the methods that we did, we used a modification of Ripley's L function, which is a multi-distance spatial cluster analysis, which basically is more robust because it's not limited by, it's not dependent on artificial partitioning of the area of interest, because as you saw, as I attempted to show you, we can just grow and grow and grow and grow and grow that circle. And also it can detect clustering, it can detect random patterns, and it can also detect regularity. And it's also robust because we use computers, and computers are way more faster than humans. They can also crunch big numbers, which we can't. So I'm hoping by utilizing this type of statistical analysis, like other researchers who have studied drill holes in the past were like, oh, if they can do it, maybe we can too, and not utilize chi-square tests. And that's kind of what I hope by when I say shift the paradigm, is just kind of like shake the old way and make them use computers to do fancier statistical tests.
Okay, we have another one from Alan Verdi, who asks, since you did not get many significant results, if you had another $500,000 to use for your next several years, what would you do differently, or what experiments would you do?
Ooh, that's a fun question. I actually would really like to continue this. That was the hope for this summer, and then things happened. Yeah, I would love to expand this experiment. I had a really fun time with it. It would be, I also think it would be really cool to just get more octopuses and see if we can maybe try it on different prey species and see if we get them to drill holes and see if like these patterns are consistent with other clams or other bivalves with 2 adductor muscles, or even just find other, like maybe snails that they eat. That would be pretty fun. Really, any opportunity that allows me to go scuba diving to collect octopuses and then run a sweet ocean acidification would be pretty cool. Ocean acidification system.
Okay.
Oh, I forgot. Sorry.
Go ahead.
Yeah, I forgot to say thanks to the scuba dive team. So for those of you who are still listening, thank you to the scuba dive team.
Okay. I'm going to ask you one more, because honestly, we could go on for a really long time, because there's actually, I've only got to about like a half of all the questions that came in. So I have to cut off somewhere, because we have another section to go over, which is JoMarie and myself and the committee are going to retire to a virtual room somewhere in cyberspace to have a deeper conversation about the written document and her presentation about the research that she did. But for now, I'll ask you this one last question, and then we will say goodbye to all the wonderful people that came.
Sure, I want to take the time to thank everybody for being here, but also for those who did send in questions and I wasn't able to answer them. Since you emailed them to Kurt, I'll just have them all forward them to me, and then I will answer them individually, because after tonight I will have a lot of time on my hands.
Okay, so Dustin asks, were the octopuses in a similar state of health? Were any senescent or sick, or were there any that weren't as healthy? Could individual health account for any differences in your data?
I think individual health did account for differences in my data, and I tried my best to determine which octopuses were senescent or gravid, or even just like missing a bunch of arms, whether or not to use them in my treatments. And there were a few, like I said, 2 or 3, and really maybe my ability to, and it probably does play into this, that my ability to detect, to recognize which octopus is healthier than the other isn't maybe as great as, say, Kurt's or Monica's and Katie's, or maybe even you, Dustin, who've handled a lot of octopuses. But I based it on what they had explained to me, and also just if they weren't eating while in acclimation, they weren't going to get used for the experiment.
Okay, with that, thank you so much, JoMarie. Thank you to everyone that came to watch this. This was, for me, a big experiment. I've never done, been a part of anything like this, a virtual thesis defense. But hey, let's all give JoMarie a round of applause for. I wish we could all hear everyone. Yeah, you did very good. Thank you very much. Now we will retire away for the second part of this this meeting. Committee members, those of you that are out there, the meeting has been set up, and so in Teams you should be able to get into there. Let's take about 10 minutes and then jump into there, so about 7:40. And again, thank you all for really sending in questions and doing that since I screwed up the Q&A functionality. Thank you for emailing them into me. I really appreciate it, and thank you for coming to attend this. Y'all have a good night.